Okay, everyone, we're going to try to keep it on schedule. I think Matt did a really nice job sort of introducing us to the stuff that you'll be seeing throughout the whole session. And so hopefully you'll appreciate why we started with a session on protein structure, because it's just going to keep coming back again and again in all the tools that we're going to do. This session, we're going to focus on how you might actually try to understand the properties of a protein using some very fundamental online tools. And so the whole premise behind this camp, is that you have the opportunity to practice and get experience using the websites that people like Matt and me use all the time in our research. And the sad thing is there's just not enough time in a standard biochemistry course to teach everybody, all the tools that they would need in addition to all the fundamental biochemistry. And so we're going to have kind of a mix here. This session is going to focus a lot more on computational tools. The session that Matt did is going to focus a lot more on standard biochemistry. But the general thinking behind this, many of you are undergrads in a research experience, and you will have a protein that you're working with throughout the summer or into the future if you're going to be around in the same lab for a while, where you'll want to understand how that protein behaves. And if your advisor or your PI asks you, hey, tell me about the molecular weight of this protein, how would you do that, right? And so the idea is that we're going to try to give you the experience that you need to say, okay, here's a new protein I'm working with. How do I find out some basic properties or some basic function, structure? How would I get to some of that information that Matt was talking about in the last presentation? And so this session is entitled Digitally Assessing Protein Properties. We're going to basically walk through a couple of websites. On your website, on the Biochemistry Bootcamp website, there's an example sequence that we're going to be using. You can also copy and paste it if you have the PDF open on your laptops. The best way to work through this session, I think the same goes with what you guys were doing with Matt, is to have those notes open, but then work along with me. So if I open up a website, go to that website and see what take a look at some of the results yourself. That will be much more easy for you to solidify what we're talking about than just trying to absorb all this material and then come back to it later on. Because this, as much as it's not an ideal format, it's a lot of lecture, but in reality, you're drinking from a fire hose here. And it's going to be very easy, unless you jot some notes down and have some sort of hands-on experience doing some of this yourself. It's going to be very easy to forget and say, oh yeah, I think we covered that in Biochemistry Bootcamp, but not remember. That's one of the big advantages of being able to get the videos up, is that we can do some of that. Anyway, so as Matt really nicely introduced in the last session, proteins are chemicals, and we can talk about them in terms of their primary structure, which is probably the most straightforward way to represent them as chemicals, because you actually see the bonds, right? I mean, we might draw it out as lysine, arginine, etc., but that's just shorthand for something that is an alpha carbon bonded to a nitrogen, bonded to a carbonyl carbon, bonded to a side chain, etc., right? And so proteins inherently are chemicals, and therefore they have all the same properties that protein chemical or that other chemicals might have. And so some of those might be the molecular weight. And so if I talk about a protein, one fundamental number is how much does it weigh? What's the molecular size of that protein? Another would be its chemical formula. And so we're going to be using GB3 a lot because it's a nice toy protein, but GB3 has a chemical formula if you wanted to write it out in your Gen Chem nomenclature as C274H427. Now, you would never write it this way. But in principle, what we want you to say is that there's nothing magic about how a protein is. It's just a hydrocarbon with some additional nitrogen, and in this case, a sulfur atom. And so there's nothing fundamentally different about a protein that isn't the same as any other molecule that you might have come across before in organic or Gen Chem. Proteins have a lot more unique properties. That's where we were talking about the secondary, tertiary, and quaternary structures. But fundamentally, they have all the properties that you would think of for any other chemical. They would have an isoelectric point. So all of those ionizable groups that Matt was talking about, they're going to have their own individual pKa's, but there's going to be some magic pH, maybe not a magic pH, but some identifiable pH where a protein has a neutral total charge, right? Where the positively charged groups in the protein match out or cancel out the negatively charged groups in that protein. Matt's already talked nicely about the sequence and residue composition. So, what percentage of this protein is glycine? How many tryptophans do I have? All of those are very important questions, particularly as you start working with proteins in the lab, right? And so this is a computer-focused course, but the idea is that we empower your research in the lab so that you don't have to go back to first principles to understand everything. And so if there's tryptophans in your protein, it's going to mean one thing when you try to measure protein concentrations than as opposed to if there's no tryptophans in your protein. Similarly, if you're doing any kind of spectroscopy, some types of spectroscopy are very sensitive to particular residues, and so you might want to know what residues are present in your primary sequence, where you might want to you might not care about some others, right? Solubility is a big one, and I wish I could give you a website that you could just put a primary sequence in and say this is the solubility of this protein. Sadly, there's no way to do that currently. There's some approximations that you can use, but it's a chemical property, and proteins have them too. I guarantee that if you're working with a new protein this summer, you're going to be hitting the wall of solubility at some point, because you're going to want to concentrate your protein so that you can use a fairly concentrated solution, and it's going to crash out at 10 micromolar or something like that. Structure is a big one. We have a couple of sessions here devoted to structure, including tomorrow's session on molecular graphics and visualization, and then concentration and extinction coefficient, right? And so, any molecule, you make a 1 molar solution of sodium chloride. You're not going to be able to do that with a protein, but in principle, you could do a concentration of a protein too. And so how would we get at this information? Because a lot of this is pretty much just standard stuff, right? But you don't want to be going through your Gen Chem routine where you're looking at the chemical structure and saying, oh, I can calculate the molecular weight by multiplying carbon, 274 by 12, plus 427 plus 609 or 69 times 14. You don't want to do all that, right? That would be a pain in the butt. And enough people have done biochemistry that they realize that there's some simple tools that you can use to do some of this that make your life simpler, right? And so, here's our sort of cast of character, singular, for today's lecture. And we'll incorporate more because, again, I'm going to be asking you guys, hey, what protein are you working on? What are you thinking about this summer? And so you'll have an opportunity to sort of guide this discussion as we go. But to start, we're going to talk about this GB3, which is actually, I like to say it's involved in bacterial cell warfare. And so it's involved in expression on the surface of Streptococcus. And there's the third IgG binding domain, so that's where the GB comes from, and the 3 because there's several of them. And it's designed to sort of interact with your immune system and try to screen the cell so that it can avoid detection, right? And so that's the biological function of this protein. That's not why we study it. We study it because it's really soluble, it's easy to make, and it has nice biochemical properties. And so in the sense that it's 56 amino acids, that means it's actually really nice for the NMR spectroscopy that we like to do. But we can think about the primary structure just like sort of we saw in the last session. We can represent those in terms of the 3-letter code. By convention, you always start with the N terminus and you end up at the C terminus. And so I've drawn those, NT is not an amino acid. That's just representing the N terminus or the C terminus. And it's much, much more convenient to represent this as single-letter codes. And so why do we have all these different nomenclatures for talking about proteins? Well, it's because people talk about proteins a lot. And in today's day of computers, the ability to take something like a sequence of letters, and I really like that example of kitchen versus thicken, right? To take that sequence of letters and convert it into something that has meaning, is really something that we do all the time because we use Google every day, right? I mean, I don't care. Well, maybe if you're like a Luddite and you just don't like computers or something, but then you're probably not taking this class. So we all use Google to search strings. When I make this amino acid sequence into a series of letters, it's just a string that can now be searched with all of the efficiency and all the different algorithms that computer scientists and computer software engineers have come up with over the last 30 years. And we can start doing really, really interesting things with these strings. Yeah, Chathuri, do you mind taking the mic back? There was a question.
This might be a dumb question, but why are the amino acids in groups of 10?
Oh, in this particular slide, I put them in groups of 10 because it's easier for me as a human to read.
Oh, okay.
Right? Computers can handle like tens of thousands of amino acids at once. For me, I'm kind of limited. Actually, if I would do it myself, I would probably do it for 5. So think about that. I mean, and that actually is a great sort of, it touches on the interface, right? Because we think in terms of chemistry, we think in terms of interactions, we think in terms of molecular function. Computers don't care, right? They have to be told how to think about those things. And so there's this interface between what makes sense to us and what makes sense to a computer. And we're not going to talk about that sort of, we're not going to go under the hood in this particular camp. I mean, if I had a whole semester, maybe we would have time to do that. But unfortunately, understanding how these algorithms are implemented and how you might do some of this, write some of this code, you kind of have to get into some real computer science and engineering to be able to do that. But in this case, the answer is really simple. I just prefer to look at groups of 10. And my computer is smart enough to say, if I see a space, that's not an amino acid. I'm just going to ignore that. And so that's why I've done it here. This also makes it super easy to see that there's 56 amino acids because I have 5 groups of 10 and 6. And so that's where GB3 is. Very nice, small, well-behaved domain. And the first website that we're going to look at is a website that is called ProtParam. Now, this is a very, very ubiquitous website. How many people, has anybody used this already? So a couple of people have. It's something that, honestly, the first thing that I do when I get a new protein, I plug it in here. And because I don't really often, I probably should write more stuff down in my lab notebook. I usually end up going back to this website over and over again because I just don't have that information off the top of my head or written down in a handy place. But this website is extremely useful, not because it does anything super complicated. Everything in this website that this website does, you could do yourself. But it makes it much easier so that you don't have to do it. So like calculating the molecular weight, calculating the amino acid frequencies in your sequence. Right? If you really wanted to, you could go through, oh, there's a glycine, oh, there's another glycine, there's a glycine, and I have 100 residues, so that means there's 3% glycine. Yeah, fine. If that floats your boat, I suspect your research advisor is going to be a little bit more demanding than just allowing you to count sequences all summer. And so this takes it and makes it in a nice, less than a second you get your result. But when you think ProtParam, you think about protein parameters. And these are going to be your basic molecular properties. A calculated molecular weight. And has anybody planning to do any mass spec this summer, mass spectrometry? So a couple people. So this would be very useful. You can on our mass spec here, and I suspect, as long as you have a good, well-calibrated mass spec anywhere, the numbers that you'll get from this will match up with the mass from your mass spec. It will match up if you're doing SDS page or gel electrophoresis. Right? So this is a great starting point to get your mass. It can give you an estimate of the isoelectric point, but if those of you who are paying attention notice that Matt said that proteins can shift around pKa's, and so it's going to give you an estimate of the isoelectric point based on the sort of unfolded or non-structured version of the protein, and so it might not be exact, but you can get an estimate for what pH your protein is going to have neutral charge. And then maybe not most importantly, but for us, often we're really interested in the concentration, and so it gives you a pretty good estimate of the extinction coefficient. All right, and so before we jump in and look at that website, let's just talk about, again, we have this balance of basic biochemistry versus understanding what's happening in the, in the computer side of things. If I think about molecular weight, right, there's lots of different ways to measure molecular weight. The most precise is going to be something like mass spectrometry, right? And again, we're not going to go into mass spectrometry here, but this is what some of your data might look like. And by looking at this, you can get your protein mass or your molecular mass or your peptide or your DNA mass down to, often, hundredths of a mass unit. So it's actually can be very, very precise. Not all of you will be doing this. Most of you, if you're working with proteins, though, will do SDS page, or this stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. Don't worry about that. SDS page is much, much easier. But most of you will be doing this, if nothing else, to assess the purity of the proteins that you might make over this summer. And so generally, the idea is you load your proteins up here, you apply a voltage, and you've added this chemical called sodium dodecyl sulfate, which basically gives all of your proteins kind of a negative charge. And so if I put a positive charge down here at the bottom, those proteins are going to migrate through the gel. And since the gel has pores of different sizes, the big proteins are going to be held up here near the top. They're not going to migrate very quickly. The small proteins, on the other hand, can migrate very, very rapidly. And if you have some standards here, you can use this as a means of estimating the molecular mass of your protein. And so if this standard is 5 kilodalton, for example, well, then that would tell you that whatever these bands are here, they're going to be about 5 kilodalton as well. Right? And so again, if you know the sequence of your protein and you could calculate the molecular weight, that'll let you, make better sense of data like this. And if you're expecting a protein that has a molecular weight of 5 kilodaltons, and say, for example, this one where you see these 3 bands, well, this tells you that your protein 1 isn't pure, right? There's other stuff there. But if this particular molecular weight marker is 15 kilodalton, well, then that gives you an estimate for what the sizes of the other bands are. And in the extreme case, you can cut these out and then run them in the mass spec to get a really, really precise molecular weight. But molecular weight's important, right? It affects all sorts of properties. Large molecules tend to have slow diffusion. They move very slowly in solution. Large molecules might have different properties when it comes to spectroscopies that you're studying. So understanding the molecular size is a very fundamental thing, in addition to being able to understand, purity on an SDS page gel. Residue composition is another thing that's useful to know. And so again, Matt's talked about disulfide formations. And so we're going to see this here, right? So again, this is not the mechanism. This is just the stoichiometry of what's going on. And in this case, you might want to talk about, there's disulfide bonds in my protein. And so some of those cysteines might be making covalent bonds to other cysteines. And that might have consequences in the molecular function if you're trying to study a protein with a particular function. And if you're doing something crazy, your protein might not work if the right disulfide bonds aren't made. And so you might need to be worried about, well, are these disulfides formed or are they not? Oftentimes, if you have cysteines in your protein sequence, you're not so interested in keeping them oxidized or forming this cysteine or cysteine bonded system. You actually want to keep them reduced, right? You want those cysteines to basically exist as single cysteines. And so some of you might come across these chemicals, mercaptoethanol, dithiothiazole, TSEP, right, as common reducing agents that'll keep those cysteines from forming disulfide dimers, right? And that can be really important if you have, cysteines sticking out into the solution. Remember some of those pictures of proteins that Matt showed in the last session, right? If there's a cysteine sticking out, and you might have some hydrophobic groups around it, and another protein has some cysteines with hydrophobic groups, well, then that's going to bring those cysteines together, and now you might have a dimer that you don't want, or that's not biologically realistic. And so these chemicals can help keep your protein reduced in such a way that it can last longer and you can use it for longer. Right? But if you don't know that your protein has cysteines in it, then you might not be paying attention to this. So understanding whether your protein has cysteines is an important feature. We do a lot of NMR in my group. Anybody going to be doing some NMR this summer? We've got a couple. Okay. Hopefully the people in my lab are raising their hand. But we will be doing NMR. And so NMR is one of those spectroscopies where you can exquisitely tell like what types of amino acids you have looking at the NMR spectrum. Right? And so this is a two-dimensional NMR spectrum called an HSQC. Again, we're not going to talk about it. There's a semester class in and of itself. But in this case, looking up here in sort of the north-central region of this spectrum, that's typically where your glycines appear. And so this is the HSQC of GB3, the protein we were just talking about. And not every peak in this region is a glycine, but 4 out of 6 are. And one of the things that my postdoc advisor used to say to me is if I was looking at a new protein, he would ask me, well, how many glycines are there? How many tryptophans are there? Because you can look at this northern region of the HSQC, and if you're expecting to see 15 glycines and you see 2, well, that probably means that you got some bad news. Probably means your protein's not right in the right conformation, or it's not, it might not be folded. The other thing is tryptophans are down here in the southwest region of this graph. And so if you expect to have 2 tryptophans and you don't see anything in that region, you might be out of luck again. So understanding your residue composition also has very important ties into the wet chemistry that you might be doing this summer. And then finally, the big one is extinction coefficient. And this one's so important that I have a little bit of experimental data here because I often find that students joining my lab will measure the extinction coefficient or measure a protein concentration, but maybe not measure it correctly. And so here, tryptophan has a very unique feature in that it absorbs light at 280 nanometers. And not much else absorbs down there, right? Some tyrosine absorbs a little bit there, but everything else is very, very low. And the nice thing about this is just like many of you might have done in your first semester or maybe second semester general chemistry classes, if you've ever talked about Beer's law, who's heard of Beer's law before? Okay, somebody tell me what Beer's law is. Right, so there's this relationship between absorption of photons, so how much light gets absorbed, and the concentration of the molecule that you have, right? And so you have more absorption, you have a higher concentration. And the proportionality constant, if you ignore path length, is called the extinction coefficient, right? And so if you know the extinction coefficient, you measure the absorption of your protein in a certain cuvette, you can immediately estimate what the concentration is. And so why might we want to know the concentration of a protein? Here's a good question. Why do I care? Yeah. So I can know how much I have, right? So that's a very basic, there's going to be multiple parts to this, so keep thinking. But yeah, if I just spent a week purifying a protein out of some bacterial cells, and I only ended up with a concentration of 2 micromolar, that's a lot of effort to put into a week's worth of work to only get 2 micromolar. And if my experiments that I want to do require me to use 10 micromolar at the minimum, well then I might be kind of looking at ways to optimize. That would be something that I need to think about. Let's think about protein interactions. Why might I want to know what the concentration of my protein is there? So let's say I have protein A, and let's just keep it simple. Let's suppose it interacts with a zinc ion. So how would I measure the concentration of the zinc ion, or how would I know the concentration of the zinc ion? Anybody remember formula weight? So one way to do this would be to take, okay, I'm going to take some solid zinc chloride out of a bottle, and I'm going to make up a solution of 100 millimolar zinc chloride, right? So, and hopefully you all know how to do that. If not, you might have some additional instruction that you need. But that's fine. Everybody's sort of uncertain about talking in these things, and that's fine. And I know most of you would know that. So you would just use the same principles that you would use for your general chemistry. You have the formula weight. It's so many grams per mole. If you're making 100 milliliter of solution, then you want a concentration of 100 millimolar. Well, then you could just do that calculation by factor label. Right? And so those types of things that you learn in general chemistry, the reason we teach you those in general chemistry is because they just come back over and over and over again. And so if you aren't comfortable with that, trust me, by the end of the summer you will be. But that zinc, that's easy. And maybe some of the more advanced of you might be thinking, well, I could use atomic absorption or something like that. Sure, go ahead. But for the most part, you can just make a solution of 100 millimolar zinc chloride, and perfect. Now you have a solution of known concentration of zinc. But if I'm trying to study, how that zinc interacts with the protein, right? And I have let's just make it even simpler. I have 100 zinc ions. If I don't know how many proteins I have, I'm not going to know what stoichiometry to mix those things together at. Or if I'm trying to do some kind of equilibrium expression, I'm going to need to know the starting concentration of protein, the starting concentration of zinc, and then work out my ICE table to figure out what the equilibrium concentrations of all the bound species are. Right? So if I don't know the concentration of my protein, there's really not a lot I can study it from a chemical perspective. I might be able to crystallize it, although the solution is to make it as concentrated as you possibly can get it. And certainly there are techniques that aren't dependent on the concentration. But many, many things that you'll be working with, even beyond just how much yield did I get from all this work I just put into it, is going to be really important as you do experiments. And so understanding the extinction coefficient is going to be a way that we can estimate the protein concentration. And so this is generally going to be done using a cuvette. I'll try to stick to this. So this is a nice small cuvette. This one actually can handle, I think, 20 microliters. Your cuvettes in your lab will vary. Generally, protein solutions are precious, and so you don't use the gigantic cuvettes that you might use in an undergrad lab where it holds like 3 milliliters of solution. In this case, it uses a small volume. Some of you might even have labs where you're using like 1 or 2 microliters, using like a nanodrop, for example, to measure the extinction coefficient. But the nice thing is that the extinction coefficient, once you know it, and ProtParam is a way to get it, allows you to plug that extinction coefficient into Beer's law so that if you know the absorption off of your spectrometer or spectrophotometer, you can then calculate the concentration given the extinction coefficient and the path length of the cell. Okay, so I think that's a good point for us to go into ProtParam and start looking at this and sort of seeing what it looks like, right? And so hopefully this switches over nicely. Looks like it did. And so one of the nice things about if you have your slides open, you can just go back and click on them. Or in my case, I guess, copy and paste. You can also Google it. It's the first hit. And so ProtParam is a deceptively simple website, right? It basically has one field. And this is the field where you would paste your protein sequence in. It assumes that it uses one-letter codes, and so you need to have the one-letter amino acid sequence. Oftentimes, if you're working on a summer project, you will have that given to you. We'll talk about some databases here in a little bit, that if you're looking something up totally new, you'll be able to sort of go in and plug things in. But for our purposes here, we're going to assume that we can take our one-letter codes of GB3, and I'm just going to copy and paste them in. And again, spaces are fine. Spaces make it easy for us to read, but the computer doesn't care. If you put something in there that's not valid, the computer will warn you, right? So those of you who know your one-letter codes know that X is not a valid amino acid one-letter code. And so if I put X in, it's going to say, hey, look, there's an amino acid here I don't recognize. But this is a well-formed sequence. And if I hit compute parameters, it takes me up with all of this information. So we're going to go through this one by one. It's not going to take too long. Number of amino acids, again, we've just talked about this. There's 56 amino acids. So it counts. If you didn't have your sequence broken down into multiples of 10, this would be one way to quickly get out the number of amino acids. Molecular weight, again, it does that for you, right? It goes in and says, okay, lysine has 10 carbons. Glycine has 2, right? It does the molecular weight calculation. And so, again, this is accurate enough. This is using sort of the standard natural abundance. And so it's not going to work if you're doing NMR labeling or isotope labeling, but we'll talk about that in a second. But this is accurate enough that I can go into our mass spec, and it's usually going to be, if I use the high res, it'll be accurate to within 1/10 of a mass unit. If I use our low res mass spec, it's usually 1 or 2 mass units. So it's pretty darn accurate. And if you're trying to convince your research advisor that the protein you have in your test tube is what the protein sequence is that you were given, mass spec is a great way to do that. Because if you get that kind of agreement, there's really not much else it can be. It gives you all of that amino acid composition, so it counts for you. And then here's where it would see these non-standard. It would say, there's some that I don't recognize here. It gives you the total positively and negatively charged residues. Again, useful for thinking about ionic interactions. Atomic composition, so it gives that to you in 2 ways. It gives you a nice little table here of number of carbons, hydrogens, nitrogens, etc. But then it also gives you the formula weight, or the molecular formula. And so, when I showed you that slide earlier where I said this is what the formula of GB3 is, again, I didn't count. I just used ProtParam to give that to me. Here's where you can sort of use this as an isotope labeling trick, right? So for an NMR, we're often making N15 labeled amino acids. I told you that this molecular weight assumes natural abundance. But if I can somehow label my protein so that every nitrogen is N15 instead of N14, well, the only thing I have to do is take this number, 6207, and add 69. Because every one of those nitrogens is going to have an additional neutron, and now I can calculate what the N15 weight of this protein will be. Most of you won't be caring about that, but again, if you take an N15 labeled amino acid and you put it on the mass spec, you kind of want to know what mass you're going to get. Right? So this is a trick to do this. No SDS page gel is going to be able to give you that kind of resolution. So you're not going to be able to tell on an SDS gel. That's really only if you're going to be using mass spectrometry. But it does show you how this information can be very useful in getting properties of your protein that you might care about in the lab. The final one is the extinction coefficient. And so here it's given to you in units of per molar per centimeter. That's important because some labs like to use mgs per mil in terms of protein concentrations, but this is in molar. How would I convert molar concentrations to mgs per mil? What information would I need? The molecular weight. And conveniently, it's right here. So you can always convert this into a mgs per mil extinction coefficient if you want to. And then there's finally some information here that I've really never used. So yeah, no, I have no idea. So don't worry about this. And if you ever figure out, if you're ever in a situation where your lab says, oh, we use this all the time, send me an email. Because I would love to know. Okay. Oh, I think for the estimated half-life, this is based on the N-terminal sequence. So if you've taken Biochemistry 2, and I know this because I just taught Biochemistry 2, and that is the only reason I know. So don't think like professors don't know everything. This is based on the first residue, and so the proteasome signaling, how fast proteins are turned over, is actually strongly dependent on the first amino acid in the chain. And so I think that's how this is being calculated. The other stuff, I don't know. I mean, I'm sure there's an equation for the gravy stuff, but I've never used that. And I don't work in mammalian reticulocytes either. So Chris Johnson might know that. You can ask him. Okay, so that's our first tool. And so we've talked about the different ways that ProtParam can help you. More importantly, we talked about what the information is, right? And so again, if you won't appreciate ProtParam if you aren't asked to know the concentration of your protein. But knowing that 9970 number basically is a game changer, because if I look here and I say that my absorbance, now we're gonna qualify this here in a second, but if my absorbance is 1.4, well, I can take that 1.4, divide it by 9970, and divide it by 1 for 1 centimeter path length, and I can give you an estimate for that protein concentration. Now, we said that most of this is coming from tryptophan. So let's do an experiment here. Right? So this is our ProtParam for GB3. And I can dig in and I can find this tryptophan residue. Let's suppose I've done a mutagenesis, or I've changed that tryptophan. Let's just make it a glycine for simplicity. I do the computation. The molecular weight's pretty much the same. It's a little bit less. Number of amino acids is the same. Almost everything else is pretty much the same. But if you noticed here, it now warns you. It says this protein does not contain any tryptophan residues. And so that means that, remember, the primary molecular moiety that's giving rise to absorption at 280 nanometers is that indole ring from your tryptophan. If that's gone, then that means you're going to absorb much, much less light. And this is strictly coming from the tyrosine residues, of which there are 3. Now, if I go in and delete the tyrosine residues, which I guess I can do because it's a small protein, and I can find them. I'm just going to delete them here. I'm not going to change them to anything. Now I'm in a world of hurt, because there are no tryp, tyrosine, or cysteine residues in your protein, and therefore you shouldn't be using UV-Vis to quantify it. So you have some alternatives in this case, and some proteins just pathologically do not have these things. One solution is say, okay, I'm going to introduce a tryptophan residue. Be a mutagenesis, or when I buy my protein, I'm going to order it with the appropriate sequence that it has a tryptophan. That's great if you've got a lab that's done that. Another way to do is BCA assay, which is a chemical that reacts with proteins and turns purple or bluish, and is a way to estimate the concentration indirectly. And so you mix the BCA reagent with the protein, you heat it up for a bit, you let it sit for a while, and if there's a lot of protein, it turns really strongly purple, and if there's not so much proteins, it stays kind of clear or yellowish. So that's another way to measure it. The third way is a bit of a hack, but it works. And this was done by a buddy of mine. So this anthus is a Nick anthus that I worked with when I was a postdoc. And so it provides a similar interface to ProtParam, but instead of giving you the extinction coefficient at 280, which is where your protein signal would be for tryptophans, it estimates it at 205 nanometers, which is measuring the backbone absorption. right? And so as you go farther and farther down, your protein backbone, that peptide bond starts to absorb light, and you can use that, again, to estimate the protein concentration. It's not perfect, and one of the big challenges with this is that if you have buffer, like heaps or tris, or any kind of organic molecule, those are going to absorb light down there too, and it's going to make it really hard for you to measure just the protein signal. You can do a buffer blank, but oftentimes the concentrations are so high that trying to blank down at the 205 nanometer region just gives you noise. Subtracting large number from large number just gives you a whole bunch of noise. And so talk to your advisor, but this website is very, very much the same in terms of ProtParam. You just give it a sequence and it spits back some numbers to you. Now, other caveats here, right? So this is accurate to within 10%. So if your lab has an extinction coefficient that you're using, don't tell them Dr. Fitzkee said to use this one, right? Use the one that your research advisor that the rest of your group uses, because presumably they're using that for a reason. This is a great, we use this primarily as our go-to for extinction coefficients. And sometimes if I see an extinction coefficient in a paper and I say, oh, that's garbage, use the ProtParam value instead, I'll make that call for my group. But you don't want to be in a position where you're making that call, particularly if the lab has used the same extinction coefficient for the last 25 years, and you're coming in and now you're like, why are all my concentrations so different? So the uncertainty can be very high with this, but generally speaking, it's pretty good. It's not dependent on the structure to a first approximation, and so we'll talk about that here in a second. But it can be dependent on sort of local bonding, or if they're extreme cases, it might be dependent on the folded structure of the protein, particularly if you're binding some other type of group. Those absorbance values, so if you've had Gen Chem, if you've done Beer's Law, you know that absorption is best between 0.1 and 1. So try to measure your absorption or your concentration on an absorption somewhere in that 0.1 to 1 range. Watch out for aggregates. So in this case, if I were to go up here and I started to see this long tail going out here into 600, 700 nanometers, to me, that's telling me that my protein isn't soluble or it's starting to aggregate. There's something in the sample that's absorbing light. And so if you have that, that's going to be bad. It basically means that the value that you're getting here in the dotted line is going to be artificially increased by the signal that you get from scattering. And so you want to try to avoid that. And so one way is to work at concentrations where your protein isn't crashing out of solution. Another way is if you can filter these out using small filters, you can often work at those types of conditions as well. And then certainly if there's DNA, DNA absorbs at 260 nanometers, and so if your sample isn't pure, now you have a mixture of protein and DNA absorbing light, and you're not going to be able to take that read of 280 nanometers because it's going to be influenced by the absorption from the DNA. And there's a section on the DNA session where we'll talk a little bit more about that as well. Okay, but I think we've covered enough that I wanted to take a bit of a break to get to this think and discuss question. And so, for you guys sitting next to each other, or if you know people in your row, if you don't know people, just turn around and introduce yourself. But we'll take maybe 2-3 minutes, and we're going to talk about this question. So I told you earlier that the extinction coefficient really doesn't depend all that much on the tertiary structure or the secondary structure, really only the primary structure. So the linear sequence of amino acids. And so why is that important? Why is that useful? Why might we care that that's the case? And so introduce yourselves, talk a little bit, and then we'll come back in 2-3 minutes and we'll see what you guys have come up with. Okay, I sense a silencing of the din. So who wants to go first? Be brave. Madison? Okay, go ahead, Madison.
So me and Joanna came to the conclusion that it's because no matter how the protein is folded, you can still measure like the concentration and stuff.
Yeah.
Yeah.
And so that's super useful because if some of you will have proteins that are not, folded in normal conditions. You might have to do what's called refolding. And so if you're working on a protein in urea, that means you can estimate the concentration. Urea denatures proteins. So you can estimate the concentration of your protein even in sort of non-standard buffer conditions. Anybody have any other ideas? I mean, the other thing that this is really useful for is if this weren't the case, none of this computer stuff would work, right? There'd be no point. You wouldn't have a website where it's asking you for the sequence. You'd have a website where it would ask you for the entire structure of your protein. And in many cases, you don't know that, right? And so being able to just say, oh, there's 3 tryptophans in this sequence, and so therefore my extinction coefficient is going to be approximately, in that case, I think, 27,000, something like that. So being able to do that will make it much, much easier because you don't have to do all the work of structure determination in order to be able to get an estimate of your protein concentration. So really basic property, but we've spent almost half of this session talking about basic properties. We're going to move faster through these other databases, but don't forget to appreciate the basics because the basics are really important. And so other types of databases that we might care about, the NCBI, the National Center for Biotechnology and Informatics, I think is what it is. I don't know exactly. We'll probably see what it is here as soon as we click it. But this is a database that basically keeps lists of sequences. And so we would give it an input of a gene name. Maybe we want to talk about an organism, or we want to look at proteins that might have been deposited by a specific investigator. That would be the input, and then the output would be curated summaries of research, right? So it would generally give you a listing of publications that have been published on that particular protein. You can get associated diseases for that particular protein. And so I mentioned earlier that most of the time you're going to be given your sequence for this summer from your PI or from your research advisor. This would be the place where if you're like, hey, I really want to see if there's any diseases out there that pertain to pyruvate kinase or triose phosphate isomerase. You could put that into this database and start looking at results. And so again, we'll take a look here briefly at this database. So does anybody have a protein that they know that they're going to be working on yet? Yeah. GFP. Okay, so green fluorescent protein. So let's see what happens. If I search for that. And so the NCBI has a ton of sort of subsidiary databases that go along with it. And so the most direct way to get information about your protein is to go click here on the protein database, right? Or the proteins family of databases. But you can also go here into things like Bookshelf, right? And so if I click on Bookshelf, it's going to bring me to a list of information from textbooks, generally, that talk about green fluorescent protein. And so, so here's an example. Now, again, so these first 4 I'm not familiar with. This one was my textbook when I was a graduate student. So if I click here into molecular biology of the cell, this is a very common textbook. I'm sure there's new editions out by now. But you can look at all of the sections that molecular biology of the cell, or commonly called Alberts, talks about green fluorescent protein. And so, here it's mainly going to be used as a tool for studying cell function. I'm guessing that if I go into the worm book, it tells me how to label nematodes with green fluorescent proteins. But let's look at some of the other ones. So here under PubMed, and we're going to get into the PubMed database more on the last day when we talk about literature searches. But if I click here on PubMed, you can see there's 68,000 results. This is probably not sufficient that I could whittle this down into a manageable reading list. But you can look at all the recent papers where green fluorescent protein was used as a tool for studying other types of molecules. We can also go here and to click into the protein. So again, I didn't really talk about this too much, but let's go in here. And this is the database of protein sequences. And so here you can see that, oh, I don't know the sequence of green fluorescent protein. But if I look here, here's a full-scale sequence of green fluorescent proteins. It's 238 amino acids. If I click on that, it's going to give me a whole bunch of, looks like PDB IDs, which we'll get into the PDB shortly. These are all different, again, this is more journal articles that have come up talking about green fluorescent protein. There's a little bit of a description of what it does. So how does it, so again, if you knew nothing about green fluorescent protein and wanted to get a sort of quick, let's go into meeting and not look like an idiot, this is a great place to start, right? Because it'll tell you sort of the basics about this protein sequence. And then if I go down here at the bottom, eventually, there's the sequence. And so now combined with ProtParam that I just looked at, this is an extremely powerful tool because I can say, okay, there's the sequence of green fluorescent protein. Now I can figure out what the molecular weight is. I can estimate the PI. I hate to tell you, Kyle, the extinction coefficient is not going to work here because it's fluorescent, right? So it's going to absorb in that region. But if this were a non-fluorescent protein, I could get an estimate of the extinction coefficient as well. Right? And the only reason it doesn't work is because he had a very specific example of a protein that strongly absorbs light. Now there's some tips, just like if you use Google enough, you know some tips about, exclude these words or include this phrase. So one thing that's often helpful is to source your protein search or your DNA search, because this also works with nucleic acids. It's to source it from what's called the reference sequence database. And so pretty much anybody can submit a sequence into the NCBI protein database. But some of those sequences are then later curated, and they're kind of the standard reference sequence that everybody refers to as, this is what I'm talking about when I'm talking about pyruvate kinase or glucose 6-phosphate dehydrogenase, right? And so if I put one of those proteins, actually let's do HIV protease, and then I'm going to put an and here because I want to add to my search query, and I'm going to type source-db refseq, and then I tell it that that's a property that I'm searching against. So that's a filter I'm using. It didn't work at all. That's why you practice beforehand. Well, let's go for one that should work. Let's do, I believe, pin 1. And sometimes you won't find a protein in the database, right? If nobody's curated it or it's not garnered sufficient interest, now I'm sure HIV protease has, but I probably just didn't form the search query appropriately. But if it hasn't garnered sort of a standard sequence out there, you might not find it in the reference sequence library. But here I entered a protease or a proloisomerase called pin 1. And so you can see there's bringing up a reference sequences from different organisms. So yeast, what's xenopus? Yeah, I'm not a molecular biology. There's mouse, humans, I guess that one's rats, it's cows. So let's suppose I wanted to save myself some time, and I really didn't want to look at any of these other, I just want to look at humans. Well, now I can put in and human, and ORGN is the ability to search for organism. And so now it should bring up only sequences of PIN1 that are from humans. And this is the reference sequences. So these should be the most sort of well-curated. Somebody's looked at them. Somebody's agreed that those are good sequences. And the first hit is the one that I would want to use. These are other sort of regions that are homologous or potentially interacting with PIN1 that have showed up. But here, if I click on the first link, again, references to recent articles, a nice summary. Again, that's something you don't always get in something that's not a reference sequence. So the nice summary sort of tells you what it does. In this case, it even gives you information on like, you know, hey, this has two domains. The first domain is a WW domain. The second domain is the rhodamase or the proteoisomerase domain. It can tell you where phosphorylation sites are. So if your protein is post-translationally modified, it gives you ideas for where those sites are. And then once again, if you scroll down, now again, this is much shorter, but if I scroll down at the end, it gives me the sequence. And if I really want the sequence, I don't want to have to cut out all this, you know, the 161, et cetera. I can click on FASTA, and that should just highlight the sequence in all caps that I could just paste right into ProtParam. So other things that you might be caring about in the NCBI databases, so we just did reference sequences. We can filter based on the PDB, and we'll talk about the protein data bank a little bit later. We can talk about restricting journals. So if I go into PubMed, and I know your advisor might be the same as mine, but you know, if I search PubMed and open up that database, no, that's horrible. Might have to cut that one out of the video. But if I search PubMed and I let's suppose I want to find recent journal articles on PIN1. If your advisor is like mine, they're going to be like, oh, you should go back to that paper that was by Wintgens, and I think they published it in, you know, the late 90s, and you know, that's all the information you're given. And so now your task is like, well, how on earth do I find that paper, right? And so, you know, here is where PubMed can actually help you, because you can search for Wintgens, and if you're really careful or really concerned, you can put it as an author, and I can add, let's do 1990 to 2000, because again, your advisor, you know, is always off by that 1 year, right? And then I use DP for date of publication, and if I search, I didn't find anything. Well, let's find all papers published between that time. I'm probably misspelling Wintgens, but that's okay. So anyway, so now I filtered by the year 1991 to 2000. You can see that there's, oh yeah, so this is KP Liu. I think this is the first paper or on PIN1. So let's see if I can't find that. Yeah, there it is. Oh, it's 2001. See, I did it myself. Late 90s. Yeah, 2001 is late 90s, right? So you can use this as a very valuable tool to do some literature filtering. Many of you will like to use Scopus or maybe Web of Science. The great thing is that PubMed is free. And if you go to your dorm room, you go to your apartment, you're going to be able to access PubMed, whereas you might only be able to use Scopus or Web of Science on campus or in your lab. And so that's where this can be really helpful. And so we've talked about some of these, using the and, the or, the not operators. All this stuff is basically tools that you can use to sort of filter your searches. And if you want, all of the NCBI databases, whether I go here to the PubMed database, or if I go here, I can always click this advanced button, and that will allow me to build up more complicated searches. Most of these sort of, and if you don't memorize all of these sort of tricks here that I have, it will build those searches for you so that you don't have to necessarily remember all of this stuff. But it's a lot quicker if you're typing in 7 or 8 searches, or you want to quickly refine something to know what those keywords are. And so I think I'm going to save this practice because I want to make sure that we get through this whole session. But if we get some time at the end, we'll maybe do one of these. But certainly, the session ends at 5. Many of you are stuck here in Starkville. This would be something where you could potentially, I don't want to say it's homework, but if you're talking with your friends afterwards and you're like, hey, I wonder what my extinction coefficient would be for the protein I'm studying, you can work out some of this and start practicing these tools yourself. The third website that we're going to talk about is the Protein Data Bank. Again, there's going to be a whole session where we discuss what PDB files are and molecular graphics. I think that's tomorrow. But we're going to introduce it here because, again, a lot of the things that Matt showed us in that first section were structural, right? And importantly, the protein structure is ultimately what influences its function. And so if I have a protein that's structured in one way, that's what's usually conserved across evolution. And so that structure is what gives rise to the positioning of the chemical groups and the placement of atoms that allows proteins to do the chemistry. And so many times when we care about proteins, we care about their structure. Even for intrinsically disordered proteins. I forget who asked that question. But there, the structure that they have is important because it's a lack of regular structure. And so are there conformational biases? Are there different properties? Now, the protein database only has nice, well-folded, well-behaved structures. And so it doesn't have disordered proteins in it, sadly. But it's still nevertheless very useful. Again, all of these things you can just Google if you're searching for them. But the website is quicker to type in if you know it. And so when I go to the Protein Data Bank, it's a very friendly looking website. Unfortunately, it doesn't have necessarily the same simplicity that maybe ProtParam or the NCBI websites have. And so if I search, let's search for pin 1 again, just because we've been doing some stuff with it. You're going to see that there's 120 structures. I'm not necessarily going to know which one is the best. I can do some filtering. So if I want to limit myself to humans, I can go click over there and click refine, and it'll refine that structural search. We're going to talk a lot more about what these numbers mean during the PDB and molecular visualization section. But most of these are coming from X-ray crystallography, and so they have a resolution associated with them. Just like your monitor, lower resolution or a smaller number there is usually a better value. Similarly, and we would, in sort of defying logic, we usually say high resolution means a low number. But in your case, in your monitor, high resolution means a smaller distance between pixels. And so for the same reason, we would generally prefer higher resolution structures. But there's other things going on as well. And so there's lots of things going on. You can't just pick the highest resolution structure and say, oh, that's the right one. And that to me is one of the challenges that X-ray crystallography faces, is how do I assess whether this structure is better than this one? We're not going to go into all that right now. But this structure is where you would download, or this database is where you would download those three-dimensional coordinates if you wanted them. And so, this would allow you to sort of access the coordinates that we were looking at. It would allow you to use another piece of software, like we'll talk about tomorrow, to generate pretty pictures and start doing analysis. But it also stores a lot of information on how the authors collected that data, right? Because this is scientific data that we're looking at. It doesn't just go poof, and now we have a structure and it's perfect. Data always has imperfections, it has uncertainties, and so with the protein data bank is all that information in being able to try to assess, is this a good structure? Where can I trust it? Where can I, what might be a little bit more dynamic than other regions? And so if I go in here, it has a lot of the scientific data information on how that structure was made. It usually has a reference associated with that structure. And then if I go here across the top, I can access all sorts of information that I may or may not care about. Again, this session, this workshop in general, is going to sort of open the door or show you where the doors are. As you go through your REU, you'll have conversations with people in your lab. If the PDB is a major part of what you're doing, trust me, you'll have conversations about what these different things mean. You'll get to learn that. Some of this stuff takes a long time to understand how all the data is generated. And so we're not going to be able to have everything presented to you. This is a taste. My postdoc just walked through the emergency door. But we'll be okay. So PDB files are generally stored with 4-character codes. And so in this case, we can look them up as ricin, for example. And if I search ricin, which is 2AAI, you can see it brings up the crystal structure for ricin. Now, I bring this up because one of our faculty members was involved in this determination of crystal structure, Deb Milsna. And so, thinking about how ricin is structured, she was in the team that solved this structure. And if we download the actual PDB file, I can save it to the desktop. And it's not a magic file, right? There's very little in science is difficult to understand or hopelessly complex, unless you're talking about quantum mechanics. But if I look at this file, I can just bring it up in a text editor, right? And so there's nothing, don't be afraid to look behind the curtain. As scientists, we're naturally curious. And so think about how these files are storing the data that they store. In this case, it's just a whole bunch of text that basically is formatted in a very specific way. And eventually, if I scroll down far enough, I'm going to get to atom entries. And right here, these 3 columns are the 3-dimensional XYZ coordinates for that atom, right? And so this information is all stored in the PDB file. We don't usually look at PDB files directly like this. But when your molecular visualization software is trying to make a pretty picture for you, it's using these XYZ coordinates to draw, oh, there's a carbon there, there's a nitrogen there. Yes? So yeah, all the stuff that we've been talking about, you're going to hopefully see lots of connections. And I don't know which came first, IUPAC or the PDB, probably IUPAC. But IUPAC cares about naming things. But if you're going to represent some protein digitally, you need a way to name every atom. And so that's built in here. And so the CAs are going to be different from the CBs or the C deltas and this is not a gamma because there's no easy way to write a gamma in a text file, at least back when they first did the PDB. And so those atom records, those lines that start with atom in your PDB, those are going to be the most important that you usually care about. I mean, now, Matt's a crystallography probably hates for me to say that because there's a lot of work that goes into the first, third of the PDB file. But for the purpose of the experimentalists interested in how the protein's structured, we're often most interested in how the atom records are, and that's what we're going to focus on. The units are always in angstroms, and if you think about your molecular sizes, your radii are typically going to be on the order of 0.5 angstrom to 2 angstroms. And so, going in here, we looked at how we could search the PDB. And so in this case, we clicked on Homo sapiens. But if you're interested in the PIN1 molecule from mouse, click that. I mean, you can basically do all of the same kind of searching in the PDB that you can do in any database. And just as before, there's this advanced search button, and you can go wild. Again, if somebody says, oh, I think that was done by, let's say, Professor Lopez back in 2017, you can search for names, you can search for years, you can honestly search for resolution, right? So there's a lot of stuff here that you can sort of play with. Again, time doesn't permit us to go through every single feature, but now that you know that it's there, you can always go back and sort of say, okay, let's see if there are any structures here that are below 1.5 angstroms. Okay. The final database I'm not going to spend too much time on. And I basically put this here because there will be, particularly in a room this size, there's going to be some real hardcore biochemistry people. Some people who are interested in pathways. Right? And so when I think pathways, I think about the KEGG database, right? And so if you've taken, has anybody here taken Biochemistry 2, second semester biochemistry? Yeah, Blake has. So you think about, where is all that pathway information stored if I don't have access to, say, for example, my Biochemistry 2 textbook? Well, I can go onto the Kyoto Encyclopedia of Genes and Genomes, and I can start searching for some of those pathways. And we'll do a basic example here. And so let's just do something really basic. Glycolysis. And so the easiest, there's other things here that are, talking about genes involved in glycolysis. We'll see once we start looking at the pathway itself, all of the data that KEGG keeps in it. But if I click here, glycolysis, it's going to give me a map that I can then click on to expand. And when I click here, this is an interactive map. So if I want to know, hey, what's the name of the protein here that converts glucose 6-phosphate to fructose 6-phosphate, that enzyme has a number. This is called an EZ number, an enzyme classification number. But if I click that point, or even if I mouse over it, it brings up the name of the enzyme. It brings up all the details. So, glucose 6-phosphate isomerase. It talks about all of the different pathways that it's involved with. And so for those of you who are like super hardcore biochemistry, where you're like, hey, and I know Steve Mansour-Body at Auburn does some of this with very sort of exotic bacteria. This is where you can sort of start looking at, oh, how does this enzyme convert one precursor into another in the context of glycolysis? And so here's an example, if you're interested in intestinal fatty acid binding protein, you can search for that. Go back to the start. So this protein's involved in shuttling fatty acids back and forth in the blood. And so I can go here into the fat digestion and absorption pathway. And now you get a nice picture of how that protein functions in the context of all of the other reactions that are going on in the cell. And if you're, you can go back into, oh, I wonder how that relates to bile. I don't, but maybe you do. You can click that button and it takes you into that pathway. And so this is a really, it's almost like an abbreviated version of a biochemistry textbook that you can sort of go through and sort of see how your enzyme fits together with other things, particularly if you're studying enzymology. Okay, so let's think about this. So I don't want to skip over this last question because I already skipped over one section, and we're a little bit early anyway. I think we end at 10 of. So let's take 2 minutes, meet up with your partners again, and let's think about some advantages and potential disadvantages of having databases like the ones we've talked about. And then we'll come back in like I said, 2-3 minutes, and we'll see what we can summarize here. Okay, so what did you guys come up with? You can't be the same people from first time. Sorry.
If somebody just kind of puts in like their crap data that they got with really sketchy methods so they can keep getting paid or whatever, then you come along and you try to use that data for something, then you're going to have bad data and your experiment's probably not going to go well.
That's a very good observation, stated about as pessimistically as I can imagine. But yeah, no, absolutely. So one of the things is that if you're, just like when you Google, who looks at the second page of their Google results these days, right? So there's the chance that you're going to get a hit that's not going to be so helpful or not going to be, up to the same level. Right? Everybody has different standards, and even people who are well-intentioned, right? I mean, it doesn't have to be necessarily somebody who's intentionally trying to throw off the field. But if I were to try to solve a crystal structure, I would try to do the best I could, but I guarantee it's not going to be the same quality as somebody like Matt or somebody who's been doing it for decades, right? And so you have this challenge where there's a lot of potential disparities. And when I mentioned earlier about the reference sequence and how databases can be curated, if you can go with curated data, that's probably the best way to go because that means that somebody else has looked at it. So what about an advantage? I mean, let's not be so pessimistic. Shouldn't be too hard. Yeah, Kyle, go ahead.
Well, I feel like the most obvious advantage is the time factor. Whereas like you spend a big part of your scientific research career looking through print journals, you can find the same information in a couple minutes, and that can exponentially increase productivity. Yes.
And that's exactly right too. I mean, I think that's the biggest advantage of this is that, we're all scientists, we're all under pressure to perform, and that's just the modern world. And so these types of tools save you from having to do the same experiments over and over and over again. 50 years ago, somebody might have, if they're interested in carbonic anhydrase, for example, you can go back and look at the papers and you'll find several different extinction coefficients because different labs invested manpower and time and resources to determine that stuff independently. There's value to that, but I think nowadays the funding agencies, the federal government, people that pay for science, they generally don't want to see you reproducing other people's results. And so this helps prevent that because you can go and find information that saves you time in the lab and makes you a more productive scientist. And so I think that's a good place to wrap it. Our very brief summary here is that protein properties, they depend on these things that we talked about in that first session, primary, secondary, tertiary structures, and that we can use databases that are organized in such a way. And certainly these are not the only 4 databases out there. There's a ton of them. But we're trying to show you which ones are the most useful that could potentially benefit you. And then just noting that curated part. If you can, find databases that are curated and are going to be less prone to error.